You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.
Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast. I have Dr. Chen. He's an assistant professor of biomedical sciences at University of California, Riverside. We'd be talking about epigenetics and specifically when is epigenetic changes heritable, to the next generation, which is a really cool topic. So, Chi, thank you for coming.
Thank you, Richard, for inviting me. So
just basically for listeners, so can you describe what epigenetics is, and then we'll dive in?
Oh my gosh. So the first question is what epigenetic is. That is quite a big one. But perhaps let's start from the very basic, what inheritance is. That might be even more basic, I think, because inheritance, I think, is the fundamental features for life, because every life, a living organism, has to pass something to the next generation, which is heritable traits. So based on this concept, I think the most important thing is what is the hereditary information carrier. So in that, right. I think people will know that it's DNA, because everyone should know that right now. But the main question is whether DNA is the only hereditary information carrier. Right now, I think the answer is no, and there are additional things. And what is these additional things? Perhaps that's something to talk about. Some epigenetic information might also be heritable. So I think that might be the first question, the answer to the first question.
Well, okay. In terms of inheriting, so we have our DNA in the cell nucleus, but then we also have our mitochondrial DNA, which is another source of inheritance. I guess some have speculated that elements in the cytoplasm that are unseen possibly could be a source of inheritance. And then I guess epigenetic changes to the underlying DNA, some may be preserved during the process of conception or creation of the sperm and the egg. So how would you cover the landscape of possible inheritable sources?
Okay, so you mean how this information, especially from environment, that can be incorporated in a genome, or perhaps the epigenome that can be inherited. So
yeah, that's a good start. What is the method by which the environment affects you, me, or another organism and changes its genetic expression?
Okay, so let's say how to start this. Basically, let's say why DNA itself cannot do this, because DNA it's a linear sequence. And for each organism, like we say, we exposed to a specific environment. Like you say, you have a big meal and you have a trauma. You mean a mental trauma today. Whether this something can change your genome in regards to the DNA sequence. So most probably it's difficult because it's just a short exposure. And it's unimaginable that this type of thing can suddenly change your sequence of DNA. So this is why there are always this idea against epigenetic inheritance, because people believe that these traits are embedded in DNA. So this type of environmental intrusion should not change DNA sequence. But on the other hand, if this type of environmental exposure or intrusions, they do not change the DNA sequence itself, but it change the marks that are associated with DNA. And it can change the order of how gene is expressed along development. So then this can change the gene outcome, like we say, the RNA and protein, and can change a trait. And most importantly, some of these traits, some of these changed structure in DNA, or perhaps the marks in DNA, can be maintained across generation. This might enable the inheritance of acquired traits through a specific environment, like we say, a big meal, mental trauma.
Well, I mean, we have I guess, a large percent of endogenous retroviruses that have happily inserted themselves into our DNA. And it appears we have many, bacterial elements too that have been inserted. And our mitochondria itself supposedly is a symbiosis of a bacteria, into our cells. So why not being able to inherit epigenetic traits?
Yeah, for this, actually, that's a very good question. That there is a possibility as well. Actually, many of the epigenetic inheritance that's discovered so far looks like related to this retrovirus sequence. So perhaps we put it in a more basic way. Like perhaps you already know a CRISPR-Cas system, right?
Yeah.
Okay, so that is actually a acquired inheritance in some way, because when a bacteria exposed to a bacteriophage, there will be many of them are killed, but some of them survive, and they capture the signature of this intrusion, and they are incorporate them into the DNA. And the second time they expose it, they can recognize it, so they can destroy the intruder. So I think this is a type of acquired inheritance. It's an example in bacteria, but I'm not sure whether this is the right parallel to the retrovirus. But something along the evolution, the genome is constantly acquire something from the environment, and this environment signal might be transferred into something like the retroviral signal and incorporated into the genome. So that's the basic idea. I'm not sure this is the right example, the right example. But since you mentioned the retrovirus, so
well, what would be a better example then, or a different example?
Yes, because what we are working right now is about RNA. Because in addition to the DNA sequence, and perhaps in addition to the DNA marks like methylation or histone modifications that attach to a DNA, and there are other potential information carriers like RNA. Because in the RNA world, many viruses still in our days still use RNA as the hereditary information carrier. So RNA in its nature might be acting as hereditary information carriers as well. And at least in mammals, these RNAs might be interacting with the genome, and whether these RNAs can add a mark on DNA, like DNA methylation, and perhaps add a mark on histone modification. The possibilities are there, but right now the evidence is still lacking in mammals. So RNA.
What causes methylation? What causes, histone deacetylation or acetylation?
These are enzymes. There must be enzymes. Enzymes has to be added at a specific loci. And the other question is where to add this methylation. The mechanisms, there are many. And actually in lower species, like in yeast, in plants, many of these mechanisms has been very well studied. And I think especially in C. elegans, the RNA-mediated epigenetic inheritance has been prospered for some time. And in mammals, there relatively lack of evidence, I think, because the molecular mechanism is not there. But right now, according to, I think in the past 5 years or so, there are more labs have discovered that the RNAs, the sperm RNAs especially, because this is what I work on, if we expose a father, whether it's a rat or mice, for me, mostly work on mice. So if we expose a mice with certain exposure, like a high-fat diet, they can change the RNA signature that resides in the sperm. And if we extract these RNAs, which has been altered by the environment, we extract these RNAs and inject them into a healthy zygote. This can change the phenotype of the offspring. So at least this tells us something that these RNAs can have a function, and this function can be related to the paternal exposure of
well, what happens to a mouse that has a high-fat diet? What happens to its phenotype, and what kind of offspring does it typically produce?
For the exposed generation, they get obese. That is the first phenotype, and they get glucose metabolism problem, and perhaps among many. So, but these are these are signature phenotypes like metabolic disorders and obesity. So that is what we observe. And in the offspring, they also have problem in coping with their glucose level. So
it looks like a heritable epigenetic change to have a high-fat diet for the mice. And then you've even transplanted the RNA from a fat mouse's sperm into a skinny mouse, and then that mouse had children, and then its children were obese as well, who had problems with glucose regulation.
You mean we just extract the RNA from the high-fat diet father and into a healthy zygote. This zygote is fertilized by a healthy sperm and a healthy oocyte, and their offspring get metabolic disorder. Is this your
okay?
Okay.
Yeah, so that further supports the fact. Okay, gotcha. That makes sense. So, what are you cataloging? What kinds of epigenetic changes are heritable versus not heritable?
Heritable versus not heritable. You mean epigenetic changes?
Yes.
Right?
Which ones? From what I've read, supposedly most of the genetic marks are wiped away.
Yes, exactly.
In the formation of them, but some may not.
Yes. So this is the actually the major criticism coming from, because in mammals there are quite extensive epigenetic reprogramming that will wipe out most of the DNA methylation and histone marks. But one argument is that some of them might not be completely wiped out, and this is one argument. But for our research, we think there perhaps another possibility. Some of the marks it will be wiped out. Let it be. But perhaps some information are not stored in the DNA itself, or perhaps attached marks. Some information might be stored in the form of RNA. And if RNA gets transferred, and this RNA might reestablish the mark that's representing the environmental exposure. So this is what we call a reconstruction process. The RNA might be the information carrier, like a cloud. Before this information gets wiped out, this information gets uploaded into the cloud, and after which this information can be downloadable and reestablish the mark. So this is another analog.
Well, where would the RNA hang out in a sperm cell? Is it just floating in the cytoplasm, and there's a lot of RNA sitting there at all times, or where does it hang out during this process?
Yeah. Actually, it's a very good question. For now, I think many of the RNA are highly compartmentalized. This is what we believe. At least in our lab, we have discovered a specific type of RNAs called tRNA-derived small RNAs. We call them tsRNAs. And some other people call it tRNA fragments. And for these RNAs, we noticed that they not only exist in the cytoplasm, but also deeply embedded in the sperm head. So perhaps deeply embedded in the nucleus. They might be part of the architecture for the nuclear organization. We don't know. But there is one fundamental truth, that the RNA in the sperm are very little. So, and if you know the shape of the sperm, it does not have much space to carry RNA. So this is from the beginning a very controversial issue. I think before year 2000, not many people believe there are even sperm RNA exist. So only later on, with the advanced technology like microarray RNA sequencing, we are now more convinced that there are indeed lots of RNA species resides in sperm as well. But yes, please.
Given the fact that the sperm is so small, does that tell you that the RNA would have to be stored in a compact form, or a different form, or that they would be stored at all?
We believe the place they are actually stored, there might be many. Like in the tail, there still might be some cytoplasmic region, and there are mitochondria as well. Perhaps some of them are inside this mitochondria, perhaps in the tail, in the sheath, and also some of them are inside the head. Some of them perhaps inside the nucleus. And they might have many places to hide. But the central question is in what form they are stored and how they can make a fuss after they go into the embryo. So I think one possibility from our research is that this RNA might be modified. They might be modified. This can prolong their lifespan after they enter into the zygote. Because when we compare the the half-life of a synthetic RNA with the same sequence and incubate with the zygote lysate, these liquid ones, these synthetic ones, degraded much faster. But the endogenous ones can survive longer. So this is one way to prolong their lifespan and perhaps extend their action inside the embryo.
So maybe there are enzymes that mark up or methylate or alter the RNA themselves and put it into a storage state for later on, and release them and bind them, and preferentially, I guess, if you have a background amount of RNA in the cell cytoplasm, perhaps that, again, there's some mechanism that sequesters a bunch of them and then releases a bunch of them, depending on what the cell needs to do.
Yeah, there is a possibility. Yeah, which means this is why I think our lab right now is focusing on this RNA modification part. It's quite intriguing because we recently have a knockout mouse, which is we delete DMT2. This is tRNA methyl transfers, which can add a specific modification at the tRNA. And with or without this tRNA, this modification, this tRNA will become more fragile. Without this modification, the tRNA become more fragile. So I think this is just the tip of the iceberg, that the methylation can change the property of RNA. And this resultant RNA, whether they carry a methylation or not, perhaps they will have different secondary structure and thus different function. So this is just a burgeoning field, I think.
Okay. Well, again, perhaps there are enzymes that do the exact same thing to RNA as they do to DNA. They, methylate it, or they,
yes.
they alter it in some way to change its
yes.
function. And maybe what happens is maybe the cell is monitoring how many RNA fragments are out there, and they're either in a storage form or an active form. I'm just speculating here. And maybe that
there is a possibility. There is indeed a possibility, and there is the other part of the story that the other lab who also works on sperm tRNA fragments from Oliver Randall lab, they showed that some of the tRNA fragments can be obtained from the somatic tissue, and during the sperm transition during epididymis. So this is another way the sperm can gain this RNA species, whether it's biogenesis from inside or perhaps take something from the somatic cells. And these somatic cells, because they are more vulnerable to environmental changes. So this is another pathway how the sperm absorb this environmental information and possibly transfer it to the next generation.
Yeah. Interesting. So what kind of experiments are you going to run to try to elucidate what's going on?
You mean what they are doing? Actually, this is the hardcore of this field, I think, what the tsRNA can do. And I think right now, not very convincing data from any labs right now. It's my feeling that, but many labs are still trying. I think the big limitation is the scarce of the material. For example, right now we can inject RNAs into the zygote and to sequence the early embryo after the injection of the RNA. We do find some genes ups and downs, but we still do not know what's the trigger. And the different lab perhaps have different explanations. But we know there are several layers of regulation. One is transcription. Perhaps the RNAs will regulate transcription directly through the interaction with other epigenetic marks. This is one possibility. The other is that perhaps these RNAs will regulate post-transcriptional processes like the RNA stability, mRNA stability, or perhaps translation directly, which means they might hijack this ribosome machinery, because ribosome is the machine that generates protein. If some RNAs can hijack the ribosome machinery, change their property, which means if they can redirect them to translate a different set of RNA. So this is another possibility. It can amplify their effect and eventually lead to the phenotype of the offspring. So there are multiple layers of things to do, but right now we need the right technique to detect these small changes in the early embryo, because the embryo is very little. Not like the cell line, like a tissue, you can get lots of materials.
Interesting. Okay. Are you also attempting, again, to catalog what kind of changes appear to be heritable and which kinds are not? Which epigenetic changes are passed along and which ones are not?
Okay, so for now, actually, it's very difficult to say because this concerns the central question that how many traits can be acquired to the next generation. We can acquire many traits in our lifespan. We are learning every day. We expose different environment every day, but we do not know how many of them can be recorded. Like for us, it's the sperm, and for the female part, it's the oocyte. We do not know how many of them can be stored, and under what circumstances they can be stored. Right now, I think there are some window of vulnerability. There must be, and perhaps under certain circumstances, these gametes can acquire more information than others. For now, we don't know. But for now, from some conferences, we know some of the information can be acquired pretty fast. Like if you drink high glucose drinking every day in addition to your regular meal, and only during a short period of time, and your sperm can carry a very distinct signature of sperm RNA. And also, if you eat a lot of high-fat diet, perhaps you can also accumulate this abnormal RNA very quickly. But the good thing is that this type of thing might also be reversible if you have a very good diet and you exercise regularly, and after exercise, your sperm RNA do change again.
Yeah, this is very strange. Huh. How long of a latency period is needed? Let's say you're going to have a mouse that's going to breed. I don't know how often its sperm are created, but
yeah, I think yes, the cycle of spermatogenesis is relatively longer. The spermatogenesis, I think, in mouse it's more than a month, and in human it's more than 70 days. So for the spermatogenesis, which means in the testis from a spermatogonia to a sperm shape, and this sperm will further go into the epididymis to be mature. So that is about 1 week. So according to some of the current data, that if a real man, I mean human being, if they are exposed to diet that unhealthy, like drinking lots of high glucose beverages, only within 1 week or two, their sperm RNA can show dramatic changes. So this just tells us one thing, that it might not go through the whole spermatogenesis in the testis. It might be just quite transient. Perhaps these changes happens in the epididymis as some others suggested. So it can be.
Maybe there's also a feedback loop where if you have just a brief period of time where, you eat a lot of glucose or you smoke cigarettes or whatever it is, that maybe the system recognizes it and corrects for that. And it only allows chronic adaptations or chronic alterations to be passed on and be heritable.
Yes, yes. I'm not sure whether this evolution have a purpose. So whether it won't change something, but perhaps it's changed. And but the consequences we do not even fully characterized. For us, when we inject the RNA, we see elevated glucose. But many people will think this is the bad thing because the glucose get higher. But what if this mouse have other adaptive changes, like their body knows the environment is full of energy, and they can elevate the glucose a little bit. Perhaps they have other adaptations that they can be they can exercise more, and they might even live longer. This is something we do not know. So, which means right now we and many other labs only detect one, perhaps, or another phenotypes that we think is important. Perhaps there are many other hidden phenotypes we do not know yet, and how this RNA species contributes to the life quality and perhaps the lifespan of this species, we still do not know. So it can be adaptive. It can be adaptive, which means some of the changes can be adaptive and perhaps can be detrimental. We do not know. And the other interesting part is that the temperature itself can also program the sperm and can change the phenotype of the offspring. Which means if you are fertilized at the winter or in the summer, your ability to accumulate this brown fat is different. So this is from another study from University of Zurich, Switzerland. I think this is quite interesting as well.
In what ways do RNA come back and interact with the underlying DNA? Like if one RNA strand, how much could it affect the underlying DNA of an organism? Could it affect just one trait? Could it affect thousands of traits?
Yes, this is another difficult question because actually from the other species, not from mammals, many of the RNA, if they want to work, they need a lot, which means there must be an amplification system to amplify the small RNA species. So like in the plants, in the C. elegans, they have a species of enzyme called RDRP. They can amplify the small RNA pathway. So you can introduce a little bit, but it can be amplified. You can have more RNA, which means the final outcome is generated by the amplified RNA species. So, but for mammals, we do not yet discover this type of RDRP enzyme in the mammals, at least in mouse, in human. So which means this put the question that how this RNA's effect can be amplified. If they just use the original amount of sperm RNA, they are very little. So, and for the well-known mechanisms of how RNA interact with DNA or perhaps with mRNA, they need quite a lot more. So this is the mystery in the mammalian field. But as you mentioned, for other species, the RNA can interact with DNA just with their complementary sequence. But this RNA will come along with other enzymes, like, for example, with perhaps the accompanied by ago protein, and they come here with other partners, either to a DNA methyltransfers can methylate the DNA. So this is one possibility, perhaps with other enzymes that methylate the histone.
So I guess there's a lot to study. Yeah, there's also what affects RNA and what can change it or put it into different forms. I mean, are you able to even look? I mean, knowing that it's probably there, has there been any dissection or analysis of a sperm cell in a mouse to see what's happened to the RNA? How much RNA is in the cell? Where is it? What form is it in? Can that at least be characterized, or is that very difficult?
You mean the species, the sperm RNA species in the RNA, you mean?
Yes.
Okay. For now, I think this is actually an evolving story, I think, for now. Because I think in 2012, our lab has discovered there are the tRNA-derived small RNAs in the sperm. But before that, most people are focusing on microRNAs and perhaps also focusing on mRNA fragments. And so I think the story is evolving. Right now we know, in addition to microRNA, mRNA, there are many tsRNAs in the sperm. But in our recent endeavor, we actually found that the sperm also carry another type of small RNAs, which is derived from ribosome RNA. We call them ribosome-derived small RNAs or RSRNAs. So this type of RSRNAs, and actually, I'm not sure how to describe them. They are derived from the different regions of ribosome RNA. It looks like they do not like a random degradation product. It looks like they are also selectively cleaved and retained in the sperm, and their function is not known. Because our previous study, we just used either injection of sperm total RNA. We know there must be something in the sperm total RNA that can generate the phenotype. Later on, we cut the RNA from the page gel that we only choose a specific segment, like a 30 to 40 nucleotide small RNAs, and we can also see a phenotype, which means the information is embedded in this specific region. But now we see that in this specific region, there are both tsRNAs and RSRNAs. So which means there might be a code of combination. So there are many tsRNAs and RSRNAs together in this region, and we inject this region as a full into the zygote. They can generate a phenotype. So which means the combination itself might be essential. there are different TsRNA species and different RNA species. Combining together, they are conveying a signal, like a 2D barcode. You can imagine.
Right.
2D barcode, you can tamper the code a little bit, you lose the signal. It looks like there are still lots of codes there, but the code is disrupted. So it must be in the right way that convey a specific signal. At least this is what we believe. But to what extent they can carry information from the environment, how specific it can carry information, this is something we still do not fully understand.
How many different species of RNA are there, approximately?
You mean species? You mean each sequence counted as a species, or perhaps?
However you want to group them. I mean, a tRNA or the other kinds of RNA. Okay. Like, first of all, how many different major kinds of RNA are there? And then within them, I'm sure there's endless variation.
Well, I think there are, as much as people has characterized from all tissues, like microRNAs, tsRNAs, rsRNAs, link RNAs, which means long coding RNAs, and mRNAs. So many species, and some of the retroviral RNAs. I think all kinds of RNAs you can imagine. So they're all there, but how they work together to convey information. That is the difficult question. And because right now we do not have a very good model to explain this, and mostly people will question that because the RNA in the sperm are infinitesimal comparing to the reservoir of the oocyte. How could such a small thing can generate a big effect? This is the main questions coming from. And what we believe there might...
virus is a very small thing compared to a cell, but look what it does.
Yes, indeed. That's our argument. There must be an amplification system that it has to target the right target. If you hijack the very essential component, you hijacked everything. So
well, not even just the targeting. Not even just the amplification. There must be a system that orchestrates what kinds of RNA to create and in what amounts, and then how to combine them to have all kinds of effects. What's the orchestration of that?
Yes. But for now, like some of the essential proteins, RNA binding proteins, you bind them and you can make a big effect. So for now, for the sperm RNA, I think there are still very little knowledge on that part. So this is, I think many labs are still struggling, including us, trying to find how they work.
Yeah, that's crazy.
Yeah, especially there is a code involved. There are so many RNAs combining together.
Well, what do we know how RNAs are generated, to start with, and how do they die? What happens to them when they're used up? Like, do they have a life cycle? And do we know?
You mean the turnover? You mean the turnover, the biogenesis and turnover of the?
Yes, that's better terms than what I put. Yeah, but biogenesis and turnover.
Yes, that's actually a essential question for each type of RNAs. But there's one interesting part. I'm not sure whether I mentioned. Yes, perhaps not today. So in the testes, the majority of the small RNA species actually is piRNA, is PV interacting RNAs in mammals. But I think most of them are retained in the cytoplasm. But during the elongation process, which is the formation of the sperm, because the sperm, during that process, the sperm will have a very compact nucleus, and their histone are replaced by protamine. So basically, they will eliminate the cytoplasm and only have this condensed sperm head, mainly composed of nucleus. So during this process, most of the piRNAs are eliminated, and in the mature sperm, the piRNA amount is relatively smaller. And after sequencing, we find mostly it's tsRNA and rsRNAs. So this is whether this can be considered as a biogenesis, we don't know. But there is a selection process. It looks like the sperm during the maturation, they only select part of the RNAs and retain in their, whether it's in the head or in the tail. So eliminate the piRNA part and choose the tsRNA and rsRNAs. So this is perhaps this is not biogenesis, right? But this is something I need to mention. And also, you might want to know how these tsRNAs are generated. So for now, the tsRNAs are generated by cleaving tRNA. This is, I think, one of the major pathway. The tRNA get cleaved, whether it's at the middle or perhaps at the terminal, and by specific enzymes. Right now, we only know a few enzymes, like androgenin or perhaps dicer in some species, and there are others. And I'm very sure that these 2 or 3 are not the only enzyme that can cleave tRNA into dsRNAs. But for now, we know very little. And I think from the evolutionary view, perhaps the degradation pathway equals the biogenesis pathway in the ancient times. So this type of cleavage is in fact a biogenesis pathway. And also for the ribosome-derived small RNAs, because there are lots of ribosome RNAs are quite long, and some of them are thousands of nucleotides. But if they are cleaved at the right side, and this might be another way of biogenesis of a new species of RNA like rsRNA.
Yeah, I know this is a side issue, but of all the organelles in a cell, are they static? Like mitochondria, from what I know, they're created and, I guess reabsorbed all the time, or they're at least, I don't know. I mean, are the other organelles like ribosomes, how many does the cell have, and are they created over time? And, how are they created? And I don't know, is there any knowledge in that regard?
You mean how often they are created, you mean?
Yeah, in the life cycle of a cell, is it first... does a cell first come into existence through cell division with all its organelles and they never change, or organelles created and reabsorbed? Is there biogenesis of organelles during a cell's life?
Wow, you mean a somatic cell? No, it's not the sperm. You are talking about other cell.
Right, a somatic cell, but yeah. For instance, a somatic, or in a sperm, are there any structures that are, throughout the life of a sperm, are there structures that are created and then, reabsorbed and created and reabsorbed?
Yeah, for this part, you mean in cycle. But for the mature sperm, I think they are quite static, at least at that specific stage. I think for the somatic cell, that's a very difficult question. That concerns the origin of the cell, how the first cell come at place. So that's, I think that's a question I cannot answer for now, but I think it's very interesting. The other thing.
Well, but not even a first cell. Like I know it's a side question, but has scientists observed a cell throughout its life, a somatic cell, and seen the appearance and disappearance of various organelles within it? Or are they static?
Yes. Actually, many of the organelles, they contain information as well. So, like you said, mitochondria contain information, and very important information, including heritable information in the mitochondria. So, but for the sperm, I think the sperm also contains something in parallel, like a centriole. But for now, we do not know this part contain hereditary information. So that's a question I cannot answer for now. But they do contain something like organelle. Some organelle might carry some important protein that can program the cell to some extent, and perhaps to change the property of the offspring. That's a big question. But for now, I think I do not know the answer. Actually, for some of the other extra genomic information, you might know the prion, this misfolded protein that causes the mad cow disease. That one you should know, right? So for the yeast, they also carry this prion. These many prion-like proteins, they can carry the property of acquired traits in yeast, and they can help the survivalness of the offspring of the yeast. So these we can also consider them as the hereditary information carrier, that in addition to DNA. So if this is what we are expecting, whether some of the organelles and perhaps some other part of the cell can have a function to storage this hereditary information carrier, whether inherited or acquired.
Well, what this tells me is the old model of all the information we need contained in the DNA, whether just nuclear or nuclear mitochondrial, is ridiculous. It seems like the information that constitutes the cell and its workings is dynamic, and it's in many, many places within the cell, not just a few, and in many forms.
That is the view. And actually, that concerns the very basic question: what is a genome? previously we think genome is just a combination of DNA sequence. It's all the sequence we have of DNA sequence. But right now it looks like it's not that simple. There are many others. Actually, the DNA secondary structure, perhaps the structure of the chromatin, also conveys information. And if that such secondary structure information can be maintained, or perhaps can be manipulated by environmental input, we can manipulate traits without changing the DNA sequence. So, and also for the RNA, like we say, the RNA perhaps can store information and can reconstruct the information. So I think it's the genome. What is the genome? So perhaps it's a combination of all the information the cell can carry, and some of them can be read and written, and some of them are read only. Like the DNA sequence itself, it will not
makes sense.
Yeah, it's read only. But for other information, like the marks, modification marks, they can be added with the ability of an enzyme and perhaps with the component with RNA. So some of the information are written and some of the information read only. So this is how certain acquired traits can be inherited. I think that's the very basis that how epigenetic inheritance can happen.
Well, this is crazy. If you think about it, a cell contains the enzymes that would help it methylate and, change its histones, right?
Yes.
So how would a cell know to have these enzymes in it? That means the cell would have to, and I'm going to anthropomorphize it, but the cell would have to know that there will be environmental stresses that will require it to adapt, and it has those enzymes ready, or at least it can produce them on demand to accommodate adaptation.
Yeah, for now, I think it's right now we cannot say that. There is a very fundamental thing we need to know. So again, it's the epigenetic reprogramming, right? So it will wipe out most of the information. So which means perhaps it's the intention that keep the genome in a clean state that can start over. We do not need to carry everything, because that will generate the chaos. That everything you expose will be inherited. That's unimaginable.
Right.
But some of them at a specific window, some information might be deeply embedded in your genome. Now, I mean, the updated version of genome, not only the DNA sequence, perhaps it's a secondary structure, it's a chromatin structure, it's a specific modification that generates a local change. So this type of information might be deeply embedded in the cell, in the genome, and perhaps in the gametes, in the sperm. And these cannot be eliminated. They will be transferred to the next generation. But to what extent this happens, and under what circumstances, under what specific environment, this is a big question now. We do not know.
Yeah, sheesh. This is crazy. The more that we learn, it's like you open up another door and it's 10 times more complicated behind that door, and that leads to 100 more doors and 100 more doors, and it's just crazy,
Yeah. Actually, I think this idea, we are not the inventor of this idea. Actually, the phenomenon has been many years ago. I think in Drosophila, in C. elegans, many people are working on it. Right now, it catches more attention only because more evidence has been found in mammals, and this will relate to human beings directly. So this is why people become more interested. I think from the basic science, many people has been working on this for a long time. And the most another thing is that this challenging the paradigm that the information acquired can be inherited, which shows Lamarckian thing, the Lamarckian evolution, which is heretical for a long time. So it become
well, knowing what you know, what are the primary drivers of evolution? I mean, random mutation, if it's there at all, it looks like it has a small part.
Yes.
And it seems like the primary driver is adaptation.
Yes. There must be something in addition to just random mutation. That will be too slow. That will not drive the world we have right now. There must be some adaptation. So, and adaptation, to some circumstances, it might have even a direction, but the final direction is to survive, right? So like a society, I think it's quite similar. We have lots of criminals, and they sabotage, but some of the criminals get caught and we will have their signature recorded, the picture taken, and their DNA sequenced. So next time we see this person, we will know he's a criminal. So we will know how to deal with it. Perhaps we will double the guard, and perhaps even we can learn something from this criminal if they carry a tattoo that belongs to a secret society. Next time we understand the people carrying this tattoo, we know, oh, this is a potential criminal. So this is an arm race. But perhaps next time the criminal will develop something more sophisticated. They will hack into the system to change their information. So there's another round of arm race. This is how evolution goes, I think. So adaptation and change and intrusion again. So.
All right. Well, I mean, gee, this has been a great call. Mind-blowing, but a great call. So
too much focus. Sorry,
that's all right. That's all right. Where can people go to start learning more? I want to just introduce them to all these concepts because it's there's so much to go into. But where can they learn more? How can they get in contact?
Perhaps just go to my website. Just Google Chen Lab. Perhaps you already have my website.
Okay. Very good.
Okay.
Well, Chi, thank you for coming. I really appreciate it.
Thank you.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.